Hi, everyone. Thanks for coming. So nice to see so many familiar faces and nice to be back here. I can't remember the last time I presented here, but it was many, many, many years ago. So I have a completely different story for you today. And I'm going to focus on the work that we've been doing in the lab really over the past decade now, examining climate change impacts on coral reefs. And this really illustrates a progression that the research program of my lab has taken from my beginnings in fisheries science and actually my beginnings as a grad student at the Fisheries Center. And from my understanding that fisheries were the biggest impact on the world's oceans. And that understanding came from the great privilege that I had of studying with Amanda Vincent, who is here today, with the late Ransom Myers and Jeff Hutchings, and with Jeremy Jackson. So 4 giants in the world of fisheries science and conservation. And I am just deeply honored and privileged to have had the opportunity to work with them, and they really shaped my thinking about oceans. And so most of the work that I did for first 10 or 15 years of my career really focused on fisheries impacts. And that's why I started studying coral reefs. And you'll see how that influence kind of changed over the time with the onset of a massive marine heat wave. This is my lab. This is how we spent much of the past 3 years. And so I just want to acknowledge the extraordinary work of all of these people and a slew of others who kind of hacked it out through a lot of time in isolation, as I know a lot of grad students spent a lot of time by themselves through COVID. And we're all very excited to be back in person. This was our lab retreat in the fall, and so super excited to be back in person, super excited to be here today. So just to give you a quick run through of some of the other work that we do in the lab that I'm not going to delve into today. COVID gave us the opportunity to work on some collaborative papers on its impacts on fisheries. We still do work collaboratively on fisheries management. I often have students working back to my roots on sharks. And we've also done work collaboratively with Tara Martin here at UBC on salmon and other fishes in the Fraser River estuary. And COVID kept us from getting to the coral reef that I'm going to tell you about today. And so we did the classic COVID pivot that everyone's been doing, and we pivoted to kelp forests, and now have a kelp forest ecosystem program that I'm actively recruiting for. So if you're interested in kelp forests, I'll have grad student and postdoc positions coming up on that. Also wanted to just quickly mention 3 pieces of work near and dear to my heart on equity, diversity, and inclusion. The first is one that I led with Tara Martin, who, as I said, is here at UBC. And this was in response to a list that many of you may have heard of that Corey Bradshaw and Frank Korchamp put up out a few years ago, which was touted as the top 100 papers in ecology that all grad students should read. And 98 of them were by men, 97 of which were white men. And so we had something to say about that. So if you would like to know more about that and unconscious bias, you can take a look at that piece. More recently, I had the opportunity to work on a piece with Sarah Davies and Amanda Bates and a number of other amazing colleagues on how narrow a lot of the metrics by which we get evaluated, in science are really, really narrow, and how we need to take a more inclusive view, both of how we're evaluated and also of mentorship models. And so that's a really nice paper that Sarah led in PLOS Biology. And then I also have a forthcoming book chapter specifically on EDI in marine sciences, so highlighting some of the specific challenges to our field that I led with Duke PhD student Leo Gaston. And that is coming out in Larry Crowder's forthcoming book, Navigating to Solutions. Okay, so the story today, however, is one that really is driven by the rapid planetary change that we're all familiar with in this room. This is from the latest IPCC report showing that we are now at that point at 1.1 degrees Celsius warming. We're now at 1.2 Celsius warming. And it is a virtual certainty that we will hit 1.5 degrees warming sometime in the next decade. So that is locked in. And this is another way of depicting this change that many of you are probably familiar with at this point. This is Ed Hawkins' warming stripes, and it is showing the average global temperature in individual vertical bars from 1850 to almost up till present, color-coded relative to a reference period in the late 20th century, with years that were colder than average in blue shades and years that were warmer in red shades. And if you were born in the mid-70s, like I was, you were born into still a fairly cool period, relatively speaking. But for the grad students in the audience, you may have been born closer to this arrow. And so just to underscore the point that for the younger people in the audience, you were born into this already very warmed world and actually don't have a baseline of what normal temperatures were. This one goes up to 2018, but this warming trend has of course continued onwards and onwards. 2020 was touted as the joint hottest year ever recorded, tied with 2016, which is really notable because 2016 was the mega El Nino that I'm going to talk about today, and 2020 was not an El Nino year. 2021 continued being just as hot. The ocean continued its trend of being the hottest ever for the 6th year on record. And just a few weeks ago, the records came out for 2022 showing that it was the 5th hottest year on record. And in the ocean, it was again the hottest year on record. So these warming trends are just marching forward. But in addition to gradual warming, we also know that climate change is increasing the frequency and intensity of a lot of extreme climatic events. And we're, of course, all very familiar with this one, the heat dome from a couple summers ago that we all experienced here, generally without air conditioning. And these extreme events also happen in the ocean. And the largest, most recent one, at least in the tropical waters, was the 2015-2016 El Nino event, which triggered the 3rd ever global coral bleaching event. This is a signature of that El Nino sweeping across from Western South America in the red across the central equatorial Pacific. And I've circled in black there the location of my long-term field site just to show that one of its distinctions is that it lies right in the path of where a lot of El Ninos hit. And this El Nino really was the wake-up call for me as to the effects and importance of climate change in the ocean. So since that El Nino, the term marine heat waves has kind of come into use and been defined clearly. And it's clear from a lot of recent analyses that marine heat wave days have been increasing substantially. The number of days has doubled since the early 80s and is predicted to increase in magnitude many, many more times as we reach 1.5 and even more if we reach 2 degrees. It's clear that marine heat waves affect all sorts of ecosystem services. It's also quite clear at this point that there is a very distinct anthropogenic signal of them. And it's also clear that extreme marine heat waves are actually becoming the norm in the ocean, meaning that most areas of the world's oceans have experienced these extreme events in the past decade. So we know that the ocean and all sorts of ocean ecosystems are being impacted both by gradual and extreme climate change. But of course, all of their vulnerabilities are not equal. And the recent IPCC report ranks different marine ecosystems according to their climate change vulnerability. And coral reefs, probably not surprisingly, are deemed the most vulnerable to climate change. And I will say kelp are the second most, or the most vulnerable temperate marine ecosystem. So one of the reasons that we've started working on them here. So the reason that coral reefs are so vulnerable to temperature anomalies is because of the really intimate symbiosis that they exist in. So temperature anomalies of as little as 1 degree Celsius can disrupt the obligate symbiosis that the coral animal exists in with microscopic photosynthetic dinoflagellates, which are now termed Symbiodinaceae. A lot of us learned these as being called zooxanthellae a long time ago, and then they were called Symbiodinium, and they're undergoing all sorts of taxonomic revisions as our molecular techniques get more and more sophisticated. This is a nutritional symbiosis. So the symbiont is photosynthesizing and providing energy to the coral. We know that these are under a microscope morphologically indistinct. So basically looking like this. They're packed inside the coral tissues really densely under typical circumstances. But we know with next generation sequencing techniques that there are hundreds, if not thousands, of different taxa, and the taxonomy is being enormous diversity of these different symbionts, and they differ enormously in their relationship with corals. So they range basically to parasites, and they also differ in their tolerance to environmental stress. So, are we okay there? Yeah. So there's a ton of research into this area because a lot of the thinking is that the types of symbionts that corals host may dictate their outcomes to heat stress. And that's because of the process of coral bleaching. So if we start with a healthy coral, as temperature or other environmental stress increases, the photosynthetic reactions start producing toxins by the symbiont. The coral therefore ejects its symbiotic partners, and if the stress continues, can starve to death. If the stress abates quickly enough, the coral can reabsorb symbionts from the surrounding water and kind of continue on. But that really hinges on how long the stress continues. So this reestablishment of symbiosis only occurs if the stress abates, and if it persists for too long, then the corals perish. And as I said, there's an enormous amount of variability, both within coral species and across coral species in whether or not they bleach. So this is just a photo example of one of these. This is a branching coral, Posilopera, that I took on Christmas Island during the 2015-2016 El Nino. And what you can see on the right is a pretty healthy Posilopera. And then right on the left, in the exact same environmental conditions, completely bleached. So a lot of questions about why there might be this level of variability. So I'm going to show you the 2015-2016 El Nino and the heat stress that was unleashed. And this spans from January 2015 all the way through the year into April 2016. Christmas Island is right in the middle of this map. It lies due south of Honolulu, just above the equator. And what you'll see is that the heat stress landed on Christmas Island in about May of 2015. Heat stress was happening in all sorts of other places in the world, including here with the evolution of the blob. Heat stress happened on lots of other coral reefs. As I said, this was the third global coral bleaching event, but on other coral reefs, it tended to move on and then move off, whereas on Christmas, it landed in mid-2015 and then just sat blanketing the reefs in heat stress for 10 straight months. So for reference, corals can bleach after as little as 4 weeks of heat stress. After 8 weeks, we start to expect mortality, and at 12 weeks, we start to expect mass mortality. This was a tweet put out by NOAA's Coral Reef Watch program in April 2016, showing the extent of heat stress that had been documented on those reefs. So Christmas is part of the Northern Line Islands chain, and it experienced the worst heat stress out of the Northern Line Islands, and in fact broke the chart basically. So prior to this, it had been predicted that no reef on the entire planet would experience 25 degrees heating stress until mid-century. And that is exactly what Christmas experienced just during this El Nino. So it was sort of a window into what we had thought would occur far in the future. And the intensity of this El Nino has really caused Coral Reef Watch to start thinking about how we calculate degree heating weeks, because we're in a kind of new uncharted waters here where we need to think about monthly heat stresses instead of weeks. Okay, so just to take you to Christmas Island, as I said, it's due south of Honolulu, and the Hawaiian island chains are those little yellow dots up there. This is a photo from space. It's the world's largest atoll by landmass, about 150 kilometers in perimeter. It's part of the Republic of Kiribati, which is a low-lying island nation flung across the central equatorial Pacific, and it is home to the Kiribati people, who are very dependent on the reef for its resources for their livelihoods and sustenance. We started working on Christmas in 2009 and have run a coupled ecological and socioeconomic monitoring program since then, up until COVID. And through interviews with people in all of the villages about their human welfare and use of the ocean, we've quantified our 40 different monitoring sites in terms of their level of chronic local anthropogenic disturbance that they experience. So this was actually one of the main draws for me when I first went to Christmas, because again, I was very focused on understanding how people alter reef ecosystems. And Christmas's reefs range extraordinarily from ones like this, and this is like right near where the villages are, so something that you can barely describe as a reef at this point. There's been an enormous amount of dredging near this site, sewage inflow, and other forms of pollution, to reefs that look like this at the other end of the atoll. So sort of this perfect ecosystem scale experiment where we could go to one location and study reefs that really span the entire gradient of possibilities. So once the El Nino was forecasted and was coming, and it was actually predicted in advance that it would hit Christmas pretty much hardest of anywhere, we had this ecosystem scale natural factorial experiment, which was quite a beautiful thing scientifically, at least. And my idea here was that in order to understand which corals are resilient to climate change, they would be subjected to this huge heat stress event. So we have to subject ecosystems to stress in order to really study resilience. It's not a static property. And the idea was that maybe because I had a fish background, I thought about tagging the corals, that we would tag them, we would track them throughout the event, taking photos, but also taking tissue samples to sequence the symbionts. And through this, we would be able to understand why some species were winners or losers through the event. So I want to acknowledge two extraordinary students who really led the charge on all of this tagged coral work that I'll talk about a little bit later. In the talk, Danielle Clark, my PhD student, and Christina Tijan, my master's student, who has been the Christmas Island project manager for 8 years now. And then a slew of colleagues that I've had the privilege of working with, most of whom are molecular ecology experts, which is quite outside my wheelhouse, but what we've been working on for quite a number of years now. And also former students, Jamie McDevitt-Irwin and Jen Magel, who is a student here and is here today. Okay, so just to briefly show you what it looks like working on Christmas. This is really one of the few long-term coral reef monitoring programs that I know of that is not based at a field station. So we work in somewhat makeshift conditions under pretty punishing equatorial sun. There's Jen. And as I said, there's no lab. So the quote unquote lab is in our little motel room, which then gets quite wet and then gets packed up for a few hours sleep before continuing everything. Okay, so I'm going to walk you through what we saw during this massive El Nino. So this was our first expedition really into it. So this was a couple of months into the heat stress. And at that point, we were really seeing variable bleaching across species. Some had not bleached yet, others had, but it was variable amongst colonies whether or not they were bleaching. Fast forward to December 2015, we were actually not out there at that point. We were still planning the next expedition, but a recreational fisherman who also has been diving on Christmas for a number of years sent me these photos, just absolutely horrified by what he was seeing. Because what he was seeing, and he recognized this, was not just completely bleached corals, but also corals that had already completely bleached, died, and been overgrown by filamentous algae. So the white looks worse, but it's actually not. This is much, much worse. Then fast forward to 10 months of heat stress, end of March 2016. We made it back out for another expedition, and this was the reef that we were faced with. So an almost complete loss of hard coral. And when we summarized all of that, which we did through thousands of photos taken on the benthos going all the way back to 2009 and looking at the damage wrought by this marine heat wave, this is what we found. 89% loss of hard coral. around the entire island. So this is an enormous amount of change in just 10 months. And you can also see huge error bars around that before estimate. And that's because of that huge difference that there was across the local disturbance gradient before the heat wave. So one of the things that we found, well, first of all, is that reefs pretty much everywhere ended up with about 4% coral cover. And what this means is that because the reefs that were very minimally disturbed before the heat wave, they actually underwent greater losses, right? So they lost about 93% of all their corals. The ones that were at very high disturbance sites already had really suppressed coral levels, and they lost about 64%. So that suggests actually that minimally disturbed, or in other places, maybe formally protected sites actually are less resilient to heat stress than disturbed sites, right? And that sort of flies in the face of one of the entire premises of conservation biology, that protected areas, communities in them will be more resilient to stressors, right? But this has been noted many times before in coral reef ecology, to the extent that it actually has a name, the protection paradox. And it's been noted a lot of times. Notably by Terry Hughes, documenting the impacts of that same marine heat wave on the Great Barrier Reef. And just looking back across successive heat waves, he was just pointing out that during successive heat waves, the heat stress had landed in different parts of the Great Barrier Reef, but also looking across the different management zones of the Great Barrier Reef, he concluded that there were no differences in the damage to corals based upon the level of protection. So he just said protection doesn't matter, it's all exposure to heat stress. Isabelle Cote has worked on this, John Bruno has worked on this in a provocative paper entitled Climate Change, Coral Loss, and the Curious Case of the Parrotfish Paradigm, which is a very strong paradigm about parrotfish controlling macroalgae on reefs that doesn't really seem to hold up. And more recently, my new colleague at UVic, who is absolutely wonderful, Amanda Bates, wrote this paper called Climate Resilience in Marine Protected Areas and the Protection Paradox, which really is based largely around coral reef ecosystems. And so the idea here is that if we start with two areas, both of which have been damaged, so in the first panel here, you've got a coral reef that has been fairly damaged, and the idea is that if it's been damaged by something like fishing pressure, destructive fishing, for example, a lot of the really sensitive species might have already been removed from that community, right? Then if you implement protection on the upper community there and allow some time for recovery, you start to get those sensitive species coming back, which here are denoted by the coral morphotype of branching corals, which would be something like Acropora or Poecilopora. Instead, down here, you're just left with this stress-tolerant community, the idea being that then when you get a heat wave or some other big stressor, if those species have co-sensitivity, so they're sensitive to fishing and they're sensitive to heat stress, then you actually get their removal again. And basically what we saw on Christmas, this much greater loss of coral in the protected area. I am not going to be finished in 10 minutes. That's super not going to happen. Just, it's not. Are we here till 12? I'm going to keep going. I'm going to apparently pick up the pace a lot. Okay. Thought we had an hour. Okay. So this is basically what we saw. Similar thing where all of the blue shades are very vulnerable species. There was this huge distinction between the communities across the disturbance gradient prior to the heat wave. And in the heat wave, we saw the loss of almost all of those sensitive species. And so on Christmas, those were things like very large tabulate acroporas, like you see here, all of which we know would have survived the previous mega El Nino in 97, 98 based on their size. Oh, I'm like so bummed. Now I don't know what to skip over. So basically, that reef that I showed you at the beginning transformed into this. And in part what this means is that there's, despite the staggering overall losses of almost 90%, there's still this huge interspecific variability in mortality levels. And this means that we have huge winners, which were all stress tolerant species, and huge losers, which were all the competitive ones. And so you can think about, not only are we losing so much coral, but we're also really changing the entire communities. Okay. I don't want to skip over. Okay, I'm going to very briefly say, previous master students, Dominique Mauchiari, investigated the soft corals and actually found for soft corals, we lost all of them. So way more sensitive than hard corals. Jen Magel, who's here, looked at these 3D models of our permanent coral reef plots, and using structure from motion imagery and analyses quantified habitat complexity, and showed that it declined quite a bit in just a year following the event. She also then looked at the reef fish communities and documented huge losses in the reef fishes. The abundance and biomass of the entire communities dropped by about 50%, but then rebounded within a year. So suggesting that they are very close to their thermal tolerance limits, but have the ability, presumably, to move to deeper, cooler waters. Okay, so I'm going to come back to the tagged corals. We ended up tagging over 1,000 corals around the island and tracking as many as we could over time. And the point that I want to make here is that, unlike the patterns that we saw at the community level, where the minimally disturbed sites lost a lot more coral, we saw a completely different, the complete reverse pattern at the species level. Okay, so that was not true for the competitive corals, the Acroporas and Montiporas, because they just died flat out. All of them across the board, over 97% mortality, so difficult to see a pattern in that. But when you look at the stress-tolerant corals, we see this striking pattern that is opposite to the community level pattern. So when you look at individual stress-tolerant corals, which you're now seeing here, there is this strong inverse pattern where mortality increases significantly as disturbance increases, which is, I think, kind of conceptually what we would think would happen. Okay, so how did this happen? I'm going to walk you through, if I have time, maybe, otherwise maybe over beers, two of the strongest winners, Platygyra and Porites, how they did it. And the first one really centers on what I think is an ecological surprise. And I really like this paper by David Lindemeyer and colleagues that laid out how valuable ecological surprises are, because they can really be catalysts for questioning how the world around us works, which you just can't do if you're doing manipulative experiments in a lab. I'm sorry. And they also really underscore the need for long-term monitoring programs, which is excruciatingly difficult to fund. But here's a paper you can cite as to their importance. Oh, I know, because I actually think I saw Carl Walters on Zoom, and I know what I wanted to say. So back in the day, I took fisheries science with Carl Walters, and he would talk every class with no PowerPoint slides, just on the board. And he would typically tell us case studies of things, ecosystems he had been working on for decades, and how all the beautiful models that he had and how it was all going to work out. And then he would get to the end and nothing would have worked. And he would just look at us and say, Mother Nature's a bitch. And that is an ecological surprise. So Carl, I hope you can hear me. Okay, so this is the backdrop for this ecological surprise. The other students can just come in and learn. This is the heat wave, okay? So this is the heat wave that fell and was superimposed on Christmas Island. This is just showing you spanning from May 2015 through to April or May 2016. And all of these dashed vertical lines are the timing of our expeditions. Okay. Here's what took us by surprise. This is Platygyra rutensis, also known as the brain coral. This is one of our corals. It's our favorite, coral 99. It's the best. It was healthy prior to the heat wave, as you would expect. Pretty healthy prior. Bleached early on, as one would expect. And then the understanding, the paradigm in coral reef ecology is that corals can only recover symbioses once the heat stress has abated. You can see where this is going. They did not. they recovered while still subjected to heat stress. And this is, to our knowledge, the first evidence of this happening. So pretty exciting, pretty surprising to us. And I guess a little glimmer of hope as marine heat waves are becoming more prolonged under climate change. It's good to know that at least some corals can reestablish symbioses while they're still being subjected to them. This coral went on to live a long and healthy life. We've revisited it many times up until 2019. Okay, how did it do that? So to understand that, we need to delve under the hood into some of that symbiotic diversity that I alluded to earlier. And this is the latest taxonomic revision by Todd Lajeunesse and colleagues. It's broken now into 9 different clades. It looks like with whole genome sequencing, there is like a ton more, but this is what we know so far. And I'm just going to highlight 2 fairly opposing clades. Clade C, which is now known as Cladocopium and is sometimes thought of as ideal symbionts. And at the other end of the spectrum, clade D, Juristinium, which are not ideal symbionts under normal conditions because they do not provide their coral host with much energy, but they are heat tolerant. So really these strong biological trade-offs. What we saw on Christmas is that prior to the heat wave, the symbiont community in the brain corals differed completely depending on where on the disturbance gradient they were. So corals that were at those very high disturbance sites clustered separately, statistically so, quite differently than the brain corals that were at any other level of disturbance. And that's because they were all dominated by Ds, Juristinium, those stress tolerant ones, which kind of makes sense because the Cs can't take the stress. It doesn't have to be thermal stress. It could be any type of environmental stress. And so the Cs had probably already been eliminated from those ones. What we found, again, rather unexpectedly, until you think about it a lot more, is that it was actually the corals that started with Cs, the heat sensitive ones, that survived. And all of the ones that started with Ds, the heat tolerant ones, perished, which seems counterintuitive. But if you think about it, that idea and that understanding comes from very short marine heat waves, where corals would eject their symbionts and maybe not make it. If they had Ds, they could probably just hang on to them throughout the whole event. Here, that was not going to work under a situation where you're subjected to heat stress for 10 months. So what these corals did, the ones that started with Cs, this is basically an index of bleaching. So a high symbiont to host ratio means that they're pretty healthy, and as they bleached, that ratio falls. The corals that started with Cs, they bleached pretty early on. And then at some point, they recovered with Ds, actually the exact same type of Ds that were in the other ones that all died. And they continued to hang on to those Ds. And we actually know now, because we have our sequences back up to 2019, that they have continued with those new D partners. So this really interesting switch of symbiotic partners. We know that corals can switch symbiotic partners, but it's never been shown before while they're still subjected to heat stress, and that it was strongly related to the level of local disturbance that they were subjected to, which is interesting because flies in the face of the protection paradox, and also because despite the fact that almost every coral reef in the world is exposed to lots of different local anthropogenic stressors, only 10% of studies that look at heat wave impacts on reefs also consider the level of underlying local anthropogenic stress. So it's a big miss, I think. Okay, I don't know if I can tell you the last story in 1 minute, but I'll try. So the other big winner was Porites, and that's this coral here and here, but it cannot switch symbionts. It doesn't have the symbiotic flexibility that the brain coral does. So it's known as a symbiont specialist. It specializes on Cs. It really can't switch between Cs and Ds. So it was a bit perplexing to us as to how it had such high survival, variable survival, overall about 70% survival across the reefs if it couldn't switch symbionts. It's known to be a specialist not just on Cs, but on one very particular type of Cs, C15. And so the thinking was that the symbionts might not have much to do with it. So we decided to investigate if there might be cryptic diversity amongst these morphologically indistinguishable Porites. Okay, so that is genetic differences that might underlie their different survival. And this work was led by my then postdoc, Sam Starko, who some of you probably know because he did his PhD here, in collaboration with Professor Sarah Davies at Boston University and her student, James Pfeiffer. So drawing upon our tissue samples taken over 6 years and 9 different time points, we used 2B RADseq to characterize the genetic structure of these different varieties. And this revealed 3 distinct genomic lineages, which you can see here in the ordination on the left, so just falling out very differently. We've called these lineages PKIR 1, 2, and 3, so P for Porites and KIR for Christmas. And they were sympatric around the reef prior to the heat wave. So all of them were found everywhere. Okay, so those are the 3 different lineages, but did that have anything to do with survival? Turns out it did. So PKIR3, which was most distinct from the other 2, just had high mortality across the board. It didn't matter where it was, exposed to disturbance or not, most of them died, about 85%. However, for the other 2 lineages, we see that strong species-specific or here lineage-specific pattern that I showed you before, where their mortality increased with greater exposure to disturbance. Okay, so is this due to the genetics of the different lineages, or is it due to the symbionts? What I can tell you is that prior to the heat wave, those 3 different lineages had really different types of symbionts. Lineage 1 and 2 had differences, but were not that different. They associated with a number of different types of C15 symbionts. PKR3 had completely different symbionts than lineage 1 and 2. They were really specialized on this fine type of C15, C15Cu. Yeah. Oh, yeah, thank you. Wondering what all that noise was. I have 2, I'm not sure why, but. 1 through 0, okay. All right. Okay. You can also see on the ordination up there that, not surprisingly from the bar plots, that they fell out in different space. So PKR 1 and 2 were kind of on top of each other, and PKR 3 are those pink dots down to the right, completely distinct. Those lineage-specific symbioses fell apart during the heat wave, so that all of the types of symbionts that lineage 3 used to associate with were lost in the heat wave, and it actually switched symbiont types to more classic C15 ones. And so we can't say for sure whether or not it's that C15 CUs, the red ones up there, maybe are more heat sensitive. It's certainly not been thought that such fine scale taxonomic differences could have functional importance, and it looks like they do have functional importance. So I think that's really, really interesting because often we're focused at a much higher level at this C's versus D's thing. But there's obviously a lot more going on here. Thinking about the corals themselves, we did find one outlier locus that differed between 1 and 2 versus 3 that has to do with coral immunity. So it could be the coral host, it could be the symbionts. We can't say for certain without further investigation, but what we can say is that there was a distinct loss of cryptic diversity with unknown consequences for the overall community. And there was a breakdown of those really tightly co-evolved symbiont relationships. Okay, so in conclusion, I don't see anyone banging down the door yet. I've shown you that a marine heat wave that was of globally unprecedented duration resulted in about 90% loss of the hard corals or stony corals. There were associated losses in habitat complexity, greater losses in the soft corals even, and temporary decline of the reef fishes. That those stony corals exhibited vast interspecific differences in mortality. So there were winners and losers, and we can think about a lot of turnover in community composition in addition to overall loss. With respect to the protection paradox, I think that what hasn't been appreciated is that these patterns might be really dependent on biological scale. And so we can see completely different things, whether we're thinking at the community level or at the species level. And I showed you for 2 of the biggest winners, 2 completely distinct mechanisms by which they were able to survive through this heat stress. one of which had to do with regaining symbiosis while still at elevated temperatures, and the other of which was underpinned by cryptic diversity. Both of those patterns were modulated by local disturbance. And as I said, despite the ubiquity of local disturbance on coral reefs, it's something that's not often taken into account under heat waves, and I think needs to be a lot more. And so with that, I will finish, I think 5 minutes past time, but hopefully still okay. I think it really depends on the duration of the heat wave, right? So if it's a quite short one, they might be actually at an advantage. And if it's a prolonged one, then I would predict they're going to be at a disadvantage.
Yeah, can you hear me? Yeah, perfect. Yeah. Hi, I just have a general question about the reefs that you've been investigating. How are they looking now? Have you done any recovery surveys? Have corals come back, or have you experienced or have you seen any shifts towards macroalgae or soft coral, corallimorpharians, which can be also detrimental to coral reef habitats, or the hard corals, basically?
The question is whether we've seen recovery, and the short answer is that up until 3 years after the heat wave, the last time we were there was 2019, there was some recovery, both of juvenile corals and also recovery from corals that had experienced partial mortality, but as colonial animals could come back. As I said, we haven't been there since 2019 because of COVID-related travel restrictions, but we're hoping to run 2 expeditions this year. And so hopefully I have an answer about that soon.